Hello there and welcome to Just About Earth, the place where curiosity takes its shoes off, settles in and begins to wonder about everything that came before us. Tonight, we're slipping back. Not just a few centuries or millennia, not even 1,000,000 years. We're going billions, deep into a time that has no photographs, no fossils you'd recognise, and no comforting sense of modern geography. A time when the continents looked nothing like the ones we know. The air was unfamiliar, and life, if you could call it that, was mostly invisible, busy making some of the most important decisions in the history of the planet. Tonight, we're visiting the Proterozoic Eon. Maybe you've never heard that word before. It's okay, most people haven't. It doesn't show up in casual conversation. It doesn't headline museum banners or pop into school curriculums as often as dinosaurs or mass extinctions. But the Proterozoic is vast, unimaginably so. It spans from about 2.5 billion years ago to just 541 million years ago. Over half of Earth's history is tucked inside that stretch. That means if you lined up everything that's ever happened, from trilobites to tyrannosaurs to Twitter, the Proterozoic is the long, quiet prelude. But it wasn't dull, not remotely. This was the age when Earth's surface was reshaped again and again, when continents collided and cracked apart, when the sky began to fill with something entirely new and slightly toxic to many early residents, oxygen, when microscopic organisms invented photosynthesis without asking anyone's permission, when cells teamed up, merged, and accidentally created the foundations for everything that would come later, including us. The rocks from this time don't tell their stories easily. They whisper if they speak at all, but buried in those layers are traces of the first great ice ages, the first complex cells, the first experiments in cooperation, the first hints that life wasn't content staying small and simple. If the Archean was Earth's chaotic childhood, the Proterozoic was its slow, sprawling adolescence, awkward, dramatic, and filled with irreversible changes. So if you're ready to drift, let's go back to a time when the sun was dimmer, the moon was closer, and a day wasn't 24 hours yet. A time before animals, before plants, before the world had a name for what it meant to be alive. Back when Earth was still figuring itself out. No rush. We've got billions of years ahead of us, and it all begins with a world just waking up. The Earth, at the start of the Proterozoic, was already more than 1 billion years old. It had been battered by meteorites, smothered in volcanic eruptions, and cloaked in a hazy atmosphere filled with gases that would be lethal to us now. There were oceans, yes, but they weren't the clear blue pools we imagine today. They were murky, metallic, and anoxic, meaning they lacked oxygen entirely. In these early waters, iron drifted freely, dissolved in abundance, giving the seas a reddish tint in some places. The land, what little there was of it, was bare rock. No trees, no soil, no buzzing of insects or rustle of leaves, just silence, wind, and the slow work of chemistry. This was not a dead world, but it was quiet. The kind of quiet that settles in when life is small and slow and very, very busy with important things. Single-celled organisms were already thriving. They had been for a long time. Tiny, ancient forms of bacteria and archaea had figured out how to survive in extreme environments, boiling vents, salty pools, places with no light and plenty of sulfur. They weren't glamorous, but they were incredibly good at what they did. They broke down minerals, they absorbed and released gases. They built structures too small for any eye to see, and in doing so, they began to reshape the planet from the inside out. The continents were starting to take shape, though they didn't resemble anything familiar. Early supercontinents, names like Columbia or Nuna, if you go looking, were assembling. They drifted slowly across the mantle, guided by convection currents that churned in the Earth's molten interior. Plate tectonics was already in play, but it hadn't yet carved out the dramatic mountain ranges or deep ocean trenches that mark today's map. Instead, land masses were low, flat, and volcanic. Erosion hadn't yet built thick layers of sediment, and without plants to anchor the soil, any loose material was at the mercy of wind and rain. Rivers existed, but they behaved differently. Lakes formed in impact craters or in cracks where the crust had pulled apart. Everything was more raw, more direct. The elements at work without the interference of large, complicated ecosystems. Sunlight reached the planet, of course, but it wasn't quite as bright as it is now. The sun, still in its early stages of maturity, shone about 85% as strongly as it does today. That lower energy output meant the Earth had to hold on to heat more carefully, which it did in part thanks to greenhouse gases like methane and carbon dioxide. The atmosphere was thick with them. There was no ozone layer, so ultraviolet radiation poured in from space, making the surface a dangerous place for anything fragile. Life stayed underwater, mostly, tucked beneath layers of ocean that acted as a protective shield. Deep water was safer, quieter, and more stable. Meanwhile, the crust beneath it all continued to shift and evolve. Early versions of continents had begun forming long before, but during the Proterozoic, the process of craton building picked up speed. Cratons are the ancient cores of continents, the geological elders of the land beneath your feet. Once they form, they tend to stick around. Stable, thick, and resistant to deformation, they're the backbone of today's continents. Much of North America, for example, sits on a craton that first solidified during this time. Africa too. Australia. Pieces of these land masses have survived billions of years of collision, rifting, erosion, and volcanic upheaval. They are survivors from an era when the surface of the planet was still figuring out how to hold itself together. One of the more subtle transformations happening during this period was in the planet's mantle. Radioactive decay was a major source of heat, and over time, that heat drove cycles of melting and remelting, helping to create new rock types. Granites became more common. So did certain minerals that would later be important for life. Phosphorus, for instance, or molybdenum. These elements didn't know they would someday help run enzymes or build DNA, but their availability would quietly shape the possibilities of biology for the rest of time. And in the oceans, something new was stirring. Among the countless microbes, a particular group had begun to master a difficult and risky form of energy production. They were bacteria, but different from the sulfur eaters and methanogens that came before. They had learned how to use light. Not just any light, but the full, unfiltered sunlight that pierced the shallow oceans. And with that light, they cracked open water molecules to release energy. As a byproduct, they gave off something unusual: oxygen. This would turn out to be one of the most important and disruptive events in Earth's history. But for now, it was just beginning. Tiny bubbles of oxygen, invisible and irrelevant, drifting through waters that had no idea what was coming. The world was still sleeping in a way, but someone had just cracked open a window. And soon, the atmosphere would never be the same. The oceans of the early Proterozoic were unlike anything we would recognise. There was water, yes, and lots of it. Vast sprawling basins covered much of the planet just as they do now. But the chemistry, the colour, the texture of those ancient seas belonged to another world entirely. These were anoxic oceans, meaning there was no free oxygen dissolved in them. And that fact changed everything. Without oxygen, many of the biological processes we take for granted simply couldn't happen. Respiration, as most modern organisms know it, didn't exist. Instead, microbial life relied on other pathways, ones that could seem alien to us today. They metabolized sulfur compounds, they produced methane, they used iron and nitrogen in ways that seem more like chemistry experiments than life strategies. It worked, but it was slow and the returns were limited. These were ecosystems built on fermentation, not fire. The colour of the water itself may have ranged from dull green to murky red depending on location. Iron was everywhere, floating freely in its dissolved form. In modern oceans, iron is scarce, quickly oxidised and precipitated out. But back then, with no oxygen to trigger that reaction, The oceans held iron in abundance. It drifted through the water column until eventually something would cause it to precipitate out, often when oxygen-producing microbes released their waste, that triggered a chemical reaction, and the iron would rust, falling out of solution to the ocean floor in massive sheets. This is how we got banded iron formations, layered rock deposits that are still mined today. They formed over hundreds of millions of years in rhythmic cycles of iron-rich and iron-poor sediment. Each band tells a story about chemical balances, about fluctuations in microbial activity, about a planet in chemical transition. They are among the best geological records we have of the Proterozoic oceans, and they only form in these kinds of oxygen-poor, iron-rich environments. Life thrived in these early seas, but you wouldn't have seen it. There were no fish, no seaweed, no corals or crabs or even plankton in the modern sense. Life was almost entirely microbial, existing in layers and films and mats. If you had the technology to observe closely, you might find stromatolites, layered structures built by colonies of cyanobacteria and other microbes. They grew slowly, layer by layer, trapping sediment and building mineralized domes over time. Some grew in shallow tidal flats, others deeper down. Many were covered by sediment and preserved as fossils, though modern examples still survive in a few isolated spots on Earth. These microbial mats were more than just clusters of cells. They were ecosystems, complex layered communities where different types of bacteria occupied different zones depending on how much light and which chemicals were available. The top layers performed photosynthesis, while the lower layers consumed the byproducts and recycled nutrients. It was a vertically organized, self-sustaining world, packed into just a few centimeters of mud and slime. Deeper in the ocean, away from the light, other microbes dominated. Archaea and bacteria adapted to low-energy environments, often living near hydrothermal vents or buried within sediments. Their metabolisms were slow, their lives measured in days or weeks rather than hours. But they were persistent, and they played a key role in regulating the ocean's chemistry. Without large animals to stir the sediment, the seafloor was a still, dark place where chemical gradients could develop over vast stretches of time. Methane was a major player in this world. Certain microbes called methanogens produced it as a metabolic byproduct, releasing it into the water and atmosphere. Methane is a potent greenhouse gas, and its abundance likely helped keep the planet warm despite the sun's reduced output. For much of the Proterozoic, the atmosphere may have been a mix of nitrogen, carbon dioxide, water vapour, and methane, with only the faintest traces of oxygen. The interaction between these gases and the ocean's chemistry was delicate. Small changes in microbial activity could lead to major shifts in atmospheric composition. And once photosynthesis began producing oxygen in earnest, the chemistry tipped. Not quickly, not dramatically, but slowly. Over millions of years, the balance started to change. Oxygen reacted with the dissolved iron, with methane, with sulfur compounds. It disappeared as fast as it was made, but the microbes kept going. They didn't need to understand the consequences of what they were doing. They just kept performing the same chemical trick, splitting water, capturing light, releasing oxygen, and the oceans began to rust. This process didn't happen evenly across the globe. Shallow waters near microbial mats saw the most immediate change, while the deep ocean remained anoxic for much longer. Scientists refer to this layered condition as a stratified ocean. Oxygen-rich at the surface, anoxic below. Over time, as oxygen levels increased and penetrated deeper, the ocean's chemistry evolved. Sulfur began to play a bigger role. Hydrogen sulfide, a gas produced by certain microbes in low oxygen conditions, accumulated in mid-depth waters. This created regions that were both anoxic and sulfidic, lethal to most modern life, but perfect for the microbes of the time. Some researchers call these euxinic conditions. They were toxic, yes, but stable. and they may have persisted on and off for hundreds of millions of years. It's a reminder that habitability is a moving target. What would kill us today was normal back then, and what we call extreme environments now were the norm for much of the planet's early history. The oceans before oxygen were quiet, chemically complex, and biologically rich in their own subtle way. They supported life, but life that played by very different rules. They were places of slow transformations, of invisible revolutions in metabolism and energy use. And they were the stage on which one of the greatest disruptions in Earth's history was about to unfold, because eventually those oxygen bubbles started to add up. It didn't start with a bang, it didn't even start with a splash. There was no sudden moment where the oceans flipped colour or where the sky shifted overnight. The great rusting of the seas, as it's sometimes called, was a drawn-out transformation, slow, silent, and utterly world-changing. Oxygen, when it first appeared in measurable quantities, was more of a pollutant than a resource. The single-celled microbes that had been producing it, primarily cyanobacteria, didn't do it for our benefit. They were just trying to survive, to make energy more efficiently by capturing sunlight and splitting water molecules. It was a clever trick, biologically speaking, but chemically, it had enormous consequences. Free oxygen is highly reactive. It doesn't just hang around. It wants to bind with things, especially metals like iron. And there was a lot of iron. Dissolved iron floated freely in the ancient oceans, giving the water a greenish or brownish cast depending on conditions. It had nowhere else to go since oxygen hadn't been around to take it out of circulation. But now, as tiny amounts of oxygen began to accumulate, the rules changed. Every molecule of oxygen that bumped into an iron ion triggered a reaction. Iron oxidized. It rusted. Not in the flaky red-brown way we see on old cars or garden tools, but in a microscopic suspended way. The oxidized iron formed insoluble particles and began to fall out of the water column. Over time, those particles settled on the seafloor, building layer after layer of iron-rich sediment. These layers hardened into rock, and what we're left with today is banded iron formations, sheets of stone that alternate between iron-heavy layers and lighter, silicate-rich ones. They're found on nearly every continent. Thick sequences in Australia, Canada, South Africa, Brazil. They vary in thickness, in colour, in mineral content. But they all point to the same thing, a long ongoing chemical shift in the planet's oceans. The process wasn't smooth, it came in waves, periods where oxygen production increased followed by times when it slowed. Sometimes the oxygen made it far enough to oxidize huge swaths of the ocean. Sometimes it stayed local, trapped near the microbial mats that produced it. The Earth was learning to breathe in fits and starts. This rusting phase likely lasted hundreds of millions of years. And it wasn't just a pretty mineral phenomenon, it was the beginning of a cascade. As more and more iron got pulled out of solution, the oceans became chemically depleted in ways that pushed other changes. Oxygen began to persist in surface waters. It started to leak into the atmosphere. Methane, once a dominant greenhouse gas, reacted with it and broke down. That had a cooling effect, which would matter later. But even before that, the presence of free oxygen created entirely new pressures on life. For many anaerobic organisms, oxygen was toxic. It damaged their enzymes, interfered with their metabolism. For a time, large portions of the biosphere were poisoned by the very gas we now depend on. Entire lineages may have gone extinct, erased quietly by chemistry they couldn't adapt to. Others found ways to cope. Some microbes evolved enzymes to detoxify oxygen's harmful byproducts. Others moved deeper, into the sediment, away from the light and the new chemical threat. Evolution didn't pause, it adjusted. The rocks preserve some of this story. In places where the banded iron formations end, where the layers abruptly disappear, we see hints that the oceans had run out of iron to rust. Oxygen had done its job. It had scoured the seas clean of free iron, leaving behind a different world, one where oxygen could finally begin to accumulate in the atmosphere in earnest. This event, the Great Oxygenation Event, didn't happen overnight. It was more like a multi-act play, with quiet intermissions and occasional reversals. But it marked a turning point. The Earth's chemistry had been altered in a fundamental way. The oceans had gone from iron-rich and anoxic to something else, something that could support different forms of life, and eventually something that could sustain complexity. The colour of the ocean changed too. Without iron to give it a rusty tint, the water likely became clearer in many places. As oxygen levels increased even slightly, different microbial communities rose to prominence. Light could penetrate deeper. New ecological layers formed. From a distance, the planet still looked much the same. Blue oceans, grey continents, white clouds. But internally, things had shifted. The rusting of the seas wasn't just about minerals, it was about momentum. A change had begun that couldn't easily be undone. Oxygen had found a foothold and it wasn't going away. The next steps wouldn't be smooth. The oxygen spike led to cooling. Methane dropped, greenhouse warming declined, and Earth teetered on the edge of its first major deep freeze. But for the microbes in the shallows, things were going according to plan. They had made a world in their image, whether anyone noticed or not. And somewhere, in a quiet pool, the oxygen kept bubbling up. It's easy to overlook the importance of something so small. Cyanobacteria are microscopic. Just tiny loops and chains of cells floating or clinging to rocks, often in places you wouldn't think to look. But what they lack in size, they make up for in impact. In the early Proterozoic, they were among the most consequential organisms the planet had ever seen. They didn't roar, stamp, or fly. They didn't build anything with bones. But they changed the chemistry of the Earth, and they did it with sunlight and patience. Cyanobacteria weren't the first living things. Far from it. Life had been around for over 1 billion years by the time they rose to prominence. But they were different in a fundamental way. They had figured out oxygenic photosynthesis. This was a biochemical trick that allowed them to take in carbon dioxide and water, absorb energy from sunlight, and use it to build sugars. In the process, they split water molecules and released oxygen as a byproduct. No one asked them to do it. They didn't coordinate or plan. This wasn't innovation in the conscious sense. It was the result of mutations, trial and error, chemical opportunism. But it worked. And once it worked, it spread. Oxygenic photosynthesis was far more efficient than the earlier versions of photosynthesis used by other bacteria, which relied on sulfur or hydrogen. That efficiency gave cyanobacteria an edge. They could grow faster, colonize new environments, outcompete their neighbours. Slowly, their populations expanded. Mats of cyanobacteria formed in tidal flats, on submerged rocks, around the edges of shallow lagoons. Some floated freely, drifting in the upper layers of the sea, capturing sunlight wherever it reached. These mats weren't just loose communities. They were layered systems, often with other bacteria living beneath the cyanobacteria, feeding on their waste products or using different wavelengths of light. Together they formed stable, resilient ecosystems. They created gradients of light, oxygen and nutrients. They cycled elements and built structures. Over time, they even helped to form stromatolites, those dome-shaped layered rocks that still turn up in fossil beds today. Stromatolites were more than geological curiosities. They were the physical record of microbial activity, preserved over thousands of years. They built up centimetre by centimetre as microbial mats trapped sediments, secreted minerals, and protected themselves with slimy coatings. The result was a laminated tower of biology and geology, a slow monument to persistence. In the early Proterozoic, stromatolites were everywhere. They dotted the shallow seas, spreading across coastlines and reef-like margins. They were one of the dominant ecosystems on the planet, not in size, but in coverage. These structures represent some of the oldest visible life on Earth, and many of them were built by cyanobacteria or similar organisms. But their real legacy wasn't in the rocks, it was in the air. The oxygen released by these microbes didn't make a big difference at first. It was absorbed by iron in the oceans, by volcanic gases, by organic matter. For millions of years, the atmosphere remained mostly unchanged. But as the Great Rusting progressed, the sinks for oxygen began to fill up. Iron levels dropped, reactive gases were consumed, and eventually the oxygen had nowhere to go but up. Cyanobacteria didn't stop. They continued releasing oxygen, oblivious to the long-term consequences. They weren't trying to reshape the world. They were just running their metabolic programs. But they became agents of transformation. The oxygen they released began to build up in the atmosphere, altering the balance of gases and triggering a cascade of environmental changes. One of the early effects was on climate. Methane, a major greenhouse gas at the time, is highly sensitive to oxygen. When oxygen levels increased, methane levels fell. This reduced the greenhouse effect, contributing to global cooling. It's likely that the first major ice ages in Earth's history were linked, at least in part, to the rise of cyanobacteria and the atmospheric shifts they set in motion. Their impact on life was even more dramatic. For many anaerobic organisms, oxygen was lethal. It interfered with their cellular machinery, disrupted their energy production, damaged their DNA. Entire ecosystems were upended. Microbial communities had to retreat to deeper sediments, more isolated environments, places where oxygen couldn't reach. Others adapted. The oxygen crisis, as it's sometimes called, forced evolution down new paths. Some microbes developed antioxidant defenses. Some began to use oxygen themselves, evolving aerobic respiration, a much more efficient way to extract energy from food. This opened the door to greater complexity, faster metabolisms, new cellular structures. Cyanobacteria weren't the end goal, they were a catalyst. They created conditions that allowed eukaryotic cells to emerge and eventually multicellular life. Their presence reshaped the biosphere, the atmosphere, the chemistry of the oceans. They didn't know they were writing a new chapter in life's story, but they were. Modern descendants of these organisms still exist. They're found in ponds on wet rocks, in hot springs and deserts. They're the green scum on a stagnant pool, the slippery coating on a river stone. They're not glamorous, but they're ancient, resilient, and still running the same biochemical playbook that changed the world billions of years ago. Every breath of air you take contains oxygen that exists because of them. Not just because of what they're doing now, but because of what they did then in the Proterozoic, when they covered the Earth in thin blue-green layers and quietly began one of the most important transformations in planetary history. For something so fundamental to modern life, oxygen had a very awkward debut. It wasn't invited, it wasn't expected. And for many of Earth's earliest organisms, it was a massive inconvenience. What we now call the Great Oxygenation Event, or GOE for short, was not a party. It was, in many ways, a slow-motion catastrophe for the microbial world. It began innocently enough. Cyanobacteria, as established, had been releasing oxygen into their surroundings through photosynthesis. At first, that oxygen didn't last long. It was absorbed by iron in the oceans, by gases from volcanic activity, by decaying organic matter. It was like pouring water onto a dry sponge. The system soaked it up immediately. For millions of years, the oxygen never made it into the atmosphere in any meaningful way. It just disappeared into the chemistry of the environment. But sponges have limits. Eventually, the sinks began to fill. The iron got used up, buried as banded iron formations. Volcanic gases shifted, slowed, or got consumed. The ocean chemistry gradually tilted. And one day, though of course no one was counting days back then, oxygen started to linger. It didn't vanish. It began to accumulate in the atmosphere. This was not a sudden event. The oxygenation process likely took tens of millions of years. But in geological terms, that's a fast shift, and for many anaerobic organisms that had evolved in a world without free oxygen, it was a disaster. Oxygen is a highly reactive molecule. It damages proteins, lipids, and DNA. In small amounts, it creates oxidative stress. In larger amounts, it's toxic. For microbes adapted to reducing environments, the rise of oxygen was like flooding their world with acid. Entire microbial ecosystems collapsed or were forced into marginal environments. Anaerobic bacteria retreated to deep sediments, hydrothermal vents, and other low-oxygen niches. Some lineages went extinct altogether. The biosphere didn't die, but it reorganized sharply. Those that could adapt survived. Others disappeared, leaving only chemical traces in the rocks. On a planetary scale, the consequences were just beginning. Methane, which had been one of the key greenhouse gases maintaining Earth's warmth under the dim young sun, began to break down in the presence of oxygen. The atmosphere cooled. Reflective ice began to form at the poles and possibly spread toward the equator. Earth may have experienced one of its first global-scale glaciations, or at least a sharp climatic downturn. This interplay between biology and climate, between microbial metabolism and planetary temperature, marked a new stage in Earth's evolution. The planet had begun to regulate itself in a way that was dynamic, interconnected, and occasionally a little brutal. The oxygen also changed atmospheric chemistry. Ozone began to form in the upper atmosphere, a molecule made of 3 oxygen atoms. Ozone is unstable near the surface but useful higher up, where it absorbs harmful ultraviolet radiation from the sun. Before the GOE, life could only survive underwater or deep in rocks, because UV radiation at the surface was intense and unfiltered. But with ozone slowly building up, the surface began to soften, becoming less hostile to life's next steps. This created a feedback loop. More oxygen meant more ozone. More ozone meant less UV radiation. Less radiation meant safer conditions for shallow water and eventually surface life. The GOE, though rough in its early phases, laid the groundwork for later biological expansion. But it wasn't all upward and onward. After the initial rise in oxygen levels, the atmosphere likely entered a period of instability. Oxygen levels rose and fell in pulses. There may have been feedback crashes, where new biological innovations or geological events caused oxygen to drop again. The early Proterozoic atmosphere was a patchwork of gases, not a stable mix. And for organisms trying to survive in it, adaptability was everything. The term oxygenation sounds pleasant, almost clinical. But to many of Earth's earliest life forms, it was more like a slow poisoning followed by a long recovery. Even for the survivors, it meant change. New metabolic strategies had to evolve. Some microbes began using oxygen in their energy cycles, developing aerobic respiration. This process was far more efficient than anaerobic pathways, allowing cells to extract much more energy from the same amount of food. And that efficiency opened doors. Larger cells became possible. More energy meant more complex internal structures. Repair mechanisms improved. Eventually, one lineage of microbes developed internal compartments, including one that specialized in oxygen processing, the mitochondrion. But that comes later. For now, oxygen was still finding its place. It lingered in the atmosphere, slowly rising. It seeped into soils and minerals. It created new types of chemical weathering, reshaping the surface of continents. The red beds, oxidized sedimentary rocks found worldwide, are part of this legacy. They formed when iron in soils reacted with atmospheric oxygen, staining entire rock layers with that familiar rusty hue. It's strange to think of oxygen as an environmental toxin, but in the early Proterozoic, that's exactly what it was. It caused extinctions, reshaped ecosystems, altered the climate, and left a lasting geological signature. It also made the modern biosphere possible. But at the time, none of that was obvious. The planet had turned a corner. Life was still small, still microscopic. But its waste product had changed the atmosphere, the oceans, and the direction of evolution itself. All without a plan. Just cells, floating in the shallows, soaking up sunlight, and doing what they'd always done. Breathe in carbon dioxide, breathe out oxygen, and let the rest of the world figure out what to do about it. The sky above the early Proterozoic Earth would not have looked like ours, at least not consistently. Depending on when you stood, if you could stand, on that raw, rocky surface, and depending on the gases hanging in the atmosphere at the time, you might see a sky tinted with hues that feel more like science fiction than geology. Not the familiar pale blue with its tidy horizon line, but something murkier, heavier, and shifting. The skies were in the middle of a transformation, and like everything else in the Proterozoic, it was happening in slow motion. Back before oxygen took hold, the Earth's atmosphere was rich in gases like methane, carbon dioxide, water vapour, nitrogen, and hydrogen. Volcanic emissions poured out sulfur compounds. Ultraviolet radiation from the sun came in mostly unfiltered. Without an ozone layer, there was nothing to block the incoming UV. And without oxygen in large quantities, there was no mechanism to create ozone in the first place. Methane in particular played a starring role. It's a greenhouse gas, yes, but it also has interesting effects on light. When present in high concentrations, methane can create a haze, an atmospheric soup of organic particles known as tholins. On early Earth, it's possible that such a haze filled the sky, scattering light and tinting the air with oranges or browns. It might have resembled something between a perpetual sunrise and a stormy dusk, depending on the thickness of the haze and the angle of the sun. Then oxygen arrived. As free oxygen began to accumulate, it reacted with methane. The breakdown of methane reduced its presence in the atmosphere, and with it, the hazy orange filter started to lift. The sky cleared. The scattering of sunlight shifted. And for the first time, the atmosphere began to behave in a way more familiar to us, allowing shorter blue wavelengths to dominate the visible sky. It's hard to pin down exactly when the sky turned blue, at least in the consistent way we experience it now. The change wasn't binary. There was no sudden click from brown to blue. Instead, it was gradual, a slow erosion of the old haze and a building up of the new clarity. Sunlight filtered through an evolving mix of gases. The blue we associate with a well-oxygenated atmosphere emerged over time. As oxygen levels rose, methane declined, and ozone began to form in the upper layers. Ozone, for its part, is created when ultraviolet light splits oxygen molecules, O2, into individual oxygen atoms, which then recombine with other O2 molecules to form ozone, O3. Once this process got going, it introduced a vital layer of protection high above the surface, something that had never existed before. The ozone layer absorbed much of the UV radiation that had previously bombarded the surface unchecked. For the first time, shallow water and eventually even land began to seem a little less hostile. Not safe exactly, but survivable. This protective layer also helped stabilise the atmosphere, at least in terms of radiation exposure. With less UV hitting the surface, organic molecules could persist longer. Mutation still happened, of course. Evolution didn't stop, but the environment became less immediately destructive. That change, in turn, opened more ecological space for microbes to explore. The visual impact of all this would have been noticeable if there were eyes to see it. And eventually, there would be. The clarity of the atmosphere would have changed the way light behaved on the surface. Shadows grew more defined, colours became sharper. Sunsets began to take on the fiery gradients we still see today. Not that there were viewers yet. No animals to blink against the glare or marvel at the horizon. Just microbial mats, quietly going about their chemical routines, while above them, the sky changed character. This shift also affected temperature. With less methane and more carbon dioxide, the greenhouse effect was altered. Methane traps heat more efficiently than CO2, so its decline likely contributed to a drop in global temperatures. In the long run, this cooling trend helped pave the way for the first major glaciations, ice ages that would wrap the planet in frost, possibly from pole to equator. But before the snow came, the light changed. The rocks from this period don't capture the sky directly. They can't tell us what shade of blue hovered over the first landmasses, or how sunlight played on the surface of an early stromatolite reef. But they can hint. Red beds, those iron-rich sediment layers, form when iron in soils and sediments reacts with atmospheric oxygen. Their presence tells us that oxygen had made its way into the air in meaningful quantities, enough to alter the way rocks weathered, enough to leave a permanent colour signature in the geological record. It's strange to think that sky colour is tied to chemistry. We take it for granted. We glance up and expect blue during the day, red at sunset, black scattered with stars at night. But that entire palette is a product of atmospheric composition, of gas mixtures and radiation angles, of molecules reacting to light. In the Proterozoic, the sky was still deciding what it wanted to be, and slowly it settled into a look we would later come to call normal. Meanwhile, down below, life continued. The microbial world didn't care much about the aesthetics of the upper atmosphere, but it benefited from the changes all the same. Oxygen wasn't done yet. It was still climbing. And with it came pressure, adaptation, and eventually a whole new kind of cell. They didn't have blueprints. They didn't plan. No scaffolding, no tools, no architects in the usual sense. And yet, these ancient microbial mats were the first communities to leave lasting marks on the landscape. Long before coral reefs or termite mounds or human cities, mats of bacteria were building something surprisingly sophisticated. A cooperative living structure, layer upon layer, cell by cell. They spread across shallow seafloors, tidal flats, lagoon edges, and coastal platforms. Wherever sunlight could reach and water didn't churn too violently, these mats found a foothold. They were thin at first, just sheets of photosynthetic bacteria clinging to surfaces, held together by mucus-like secretions. But over time, the mats grew more complex. They developed vertical structures, stacked communities, gradients of light, oxygen, nutrients, and waste. It was zoning by chemistry. The top layers were dominated by Cyanobacteria. They were the ones doing the heavy lifting, capturing sunlight, splitting water, producing oxygen, and synthesizing sugars. Just beneath them were other microbes, anaerobes, that used the Cyanobacteria's waste products, hydrogen sulfide users, nitrogen fixers, fermenters. Everything had its niche. The deeper you went, the less oxygen there was. And in those low oxygen or anoxic zones, the microbial metabolism shifted. Instead of generating oxygen, these organisms broke things down. They recycled, they decomposed, they turned organic material back into usable forms for the layers above. It wasn't a hierarchy, it was a loop. Some of these mats trapped sediments, tiny grains of sand and silt carried in by tides or currents. Others precipitated minerals out of the water using their metabolic byproducts. Over time, these processes built up layered structures, flat at first. Later, more domed or columnar. These became stromatolites. Some formed isolated clumps. Others spread for kilometres, covering entire coastal shelves in microbial patchwork. Stromatolites are still found in a few places today. Shark Bay in Australia, some hypersaline lagoons, a few alkaline lakes. Modern examples grow slowly, often in harsh conditions where grazing animals or modern seaweeds don't interfere. They're echoes of a time when these structures were everywhere. In the Proterozoic, stromatolites were among the dominant surface features of the shallow ocean, not because they were tall or fast-growing, but because there was so little competition. Nothing else built permanent mineralized structures on this scale. Nothing else engineered its environment in such a visible, layered way. These weren't just biological communities, they were geobiological machines. Each layer of a microbial mat performed specific functions, and the entire structure regulated its internal chemistry. Oxygen was produced in one place and consumed in another. Sulfide was generated in one layer and used in the next. Carbon cycled through photosynthesis, respiration, and fermentation. It was dense with activity, even if it looked like little more than a greenish slime from the outside. The mats altered their surroundings. They influenced sediment stability, trapping grains and building microenvironments. They changed the composition of nearby waters, altering pH and redox conditions. They were, in a way, tiny cities. Systems with neighbourhoods, specializations, waste management, energy generation, and defense. There were no predators, not yet. But competition still existed. Different strains of bacteria competed for space, for light, for access to nutrients. Some mats produced antimicrobial compounds to keep out invaders. Others grew faster or adapted to slightly deeper waters. Evolution didn't wait for eyes or limbs to get clever. The architecture wasn't made of stone or bone, but it was durable. Stromatolites preserve well, especially in carbonate-rich environments. The layers lithified over time, turning into rock. Some preserved microfossils, tiny filaments and spheres that hint at the cells that built them. Others preserved only textures. But either way, the record is there. These structures tell us more than just who lived where. They offer clues about the chemistry of the oceans, the levels of oxygen, the intensity of light. They tell us which microbes were thriving and which environments supported complexity. They also mark time. Layers build slowly, counting them, measuring them, analysing their composition. This gives geologists a way to trace environmental changes across millions of years. There's also something quietly profound about how long these mats held their place. For most of the Proterozoic, microbial mats were the dominant ecological form. Not a side story, not a niche. They were the headline. For over 1 billion years, no other life form came close to displacing them. The slow, coordinated, layered life strategy worked. And so it persisted. Eventually things would change. New cells would arrive. Grazers would evolve. The stability of the mats would be disrupted. But that was still far off. For now, in the middle of the Proterozoic, they were the primary engineers of the biosphere, shaping coastlines, regulating chemistry, building quiet cities, one layer at a time. For most of Earth's history, life didn't have eyes. It didn't have mouths or spines or bones. There were no wings cutting through the air or fins slicing through water. There was no movement, not in the way we think of it. Nothing hunted, nothing fled. There was no seeing, no reacting to light beyond the level of molecules and membranes. And yet, life thrived. The Proterozoic Eon was a microbial world. Everything was small. Nearly everything was single-celled. The diversity was enormous, but almost all of it unfolded at the microscopic scale. Not because larger life was impossible, but because there had been no need yet for it. No arms race, no chase between predator and prey, no survival pressure that required speed, complexity, or central coordination. Microbes ruled through chemistry, not behaviour. They shaped their environment by existing, by breathing, by excreting. Every cell was a little factory, running ancient metabolic scripts. Absorb, convert, release. There were no faces to read, no limbs to wave. But there was communication. Chemical signals passed between cells, gradients that told microbes where to move, when to grow, whether to divide. They moved when needed. Some had flagella, whip-like tails that let them swim toward nutrients or away from danger. Others drifted with currents, passive passengers in water columns. Some clung to surfaces, some sank and waited. Life didn't need to be fast. It needed to be efficient. A lack of eyes didn't mean a lack of light sensitivity. Many microbes responded to light, especially those involved in photosynthesis. Cyanobacteria positioned themselves in water columns to catch just the right wavelengths. Some even adjusted their depth as light changed with the time of day or water turbidity. They had no vision, but they had photoreceptors, molecular systems that triggered changes in behavior when light hit them. The microbial biosphere was structured by gradients: light at the top, darkness below, oxygen in some places, anoxia in others, temperature, pH, salinity, all varied in layers and pockets. Microbes specialized. Some thrived only in the narrow band where two conditions overlapped. Others endured extremes: heat, salt, acidity, pressure. The variety was immense, even without visible features. This was not a static world. Genetic change was constant. Bacteria reproduce quickly, and each division carries the possibility of mutation. Over millions of years, this allowed for extraordinary diversification. Horizontal gene transfer, where microbes share genes directly, sped things up even more. Traits like photosynthesis, nitrogen fixation, or resistance to toxins could hop between lineages. Evolution didn't wait for complexity. It found every available shortcut. The fossil record from this time is limited. Most microbes didn't leave behind hard parts. Their bodies decayed quickly. But they did leave chemical signatures, biosignatures. Certain molecules, like hopanoids or steranes, point to the presence of specific kinds of bacteria. Stable isotope ratios suggest who was doing photosynthesis, who was digesting sulfur, who was fixing nitrogen. The rocks remember even when the cells themselves are long gone. Sometimes, preserved mats or sediments trap impressions of ancient microbial life. Tiny filaments, cell clusters, mineralized remains. Not photographs, but traces. Just enough to piece together a picture of a world teeming with life, even if none of it had faces. The ecosystem was self-sustaining. Primary producers, photosynthesizers, created energy-rich molecules. Consumers broke them down. Decomposers finished the job. It was a full food web just stripped of any physical drama. No jaws, no chase. Just molecules moving through layers of mud and water, slowly but endlessly. Time moved differently here. Without complex nervous systems, there was no awareness, no memory, no planning. Cells responded to their environment in real time through chemistry, a signal, a change, a reaction. That was the rhythm of life. And for all its simplicity, this system was stable. It lasted for over 1 billion years. No collapse, no extinction events as we think of them. There were shifts, sure. New metabolisms arose. Oxygen levels changed, climate fluctuated. But the microbial biosphere adjusted. It absorbed shock. It filled every niche and recycled every molecule. It's tempting to think of this time as a prelude, a slow build-up to the real action. But that's not how evolution works. There is no finish line. The microbial world wasn't waiting for eyes or legs or intelligence. It was complete in its own way, finely tuned, successful on its own terms. Many of the lineages that dominated then are still here now. Some have barely changed. Their descendants live in our guts, in our soil, in our oceans. Others were ancestral to entirely new forms of life, eukaryotes, multicellular organisms, animals. But at this point in the Proterozoic, that future hadn't happened yet. This was their time. A world shaped by cells too small to see, building empires of chemistry, surviving without sight, without sound, without anything we'd recognize as awareness. And still, they were the most powerful force on the planet. The reign of the microbes wasn't just long, it was foundational. And in one corner of this invisible world, something new had begun to stir, something with a nucleus. They didn't appear with a flash. No dramatic split, no ancient flag planted in the mud. Eukaryotes, the cells that would one day build all plants, animals, fungi, and everything complex in between, arrived quietly. Like so many things in the Proterozoic, their appearance wasn't sudden. It was gradual, messy, and wrapped in uncertainty. But when they did emerge, they marked a major departure from everything that had come before. A eukaryotic cell is more than just a membrane and some cytoplasm. It's compartmentalized, organized, internally specialized. It has a nucleus where its DNA is kept safe behind a second membrane. It has organelles, tiny structures with specific roles, mitochondria to make energy, endoplasmic reticulum to process proteins, a cytoskeleton to maintain shape and guide movement. It's not a bag of chemicals. It's a system with traffic patterns, logistics, storage, and oversight. None of that existed in earlier life. Prokaryotes, bacteria and archaea, are simple by comparison, efficient but stripped down. Their DNA floats free in the cell. Reactions happen more or less wherever conditions are right. That simplicity works. It's worked for billions of years. But eukaryotes took a different route. They opted for internal complexity. How that happened is still debated. The leading explanation is symbiosis, a kind of cellular merger. At some point, an archaeal host cell took in a bacterial partner, not as food, but as a collaborator. Maybe the bacterium started living inside the host, protected from the outside world. Maybe it offered something in return, like the ability to use oxygen efficiently for energy. Whatever the exact circumstances, the partnership worked. The bacterium stayed. It lost its independence, shed unnecessary genes, and became an organelle. Over time, it evolved into what we now call a mitochondrion. This wasn't the first time life merged, but it was one of the most consequential. Suddenly, the cell had a dedicated energy center, one that could churn out far more ATP, the molecule cells use for fuel, than any previous metabolism allowed. That extra energy made everything else possible. It allowed for larger genomes. It supported the cost of running internal compartments. It gave rise to the potential for cell specialization, for regulation, for scaffolding, for shape. It was, in evolutionary terms, a major upgrade. Not required, but powerful. The earliest eukaryotes were still single-celled. They weren't multicellular yet. They didn't have brains or limbs or recognizable features, but they had potential, and they began to diversify. Some became ancestors of algae. Others branched toward what would become fungi or animals. They took different paths, tried new forms. Many of those experiments didn't last. Evolution is full of false starts. But some lineages persisted, and over time, the eukaryotic tree filled out. What marks this period is not abundance, but innovation. Eukaryotes weren't dominating ecosystems. They were newcomers in a microbial world still ruled by bacteria and archaea. But they were doing things differently. Some developed flexible membranes and the ability to engulf other cells. This opened the door to predation, a new kind of ecological interaction. Life wasn't just absorbing molecules from the environment anymore. It was starting to feed on other life. This was a subtle but enormous shift. It created selective pressure for defense, for mobility, for detection. Eukaryotes didn't need eyes yet, but they needed awareness, chemical sensing, environmental responsiveness, the ability to move toward or away from stimuli. Life was beginning to think spatially. They also started forming more elaborate relationships with each other. Some became hosts to other symbionts. Certain groups eventually took in photosynthetic bacteria, likely Cyanobacteria, and those guests became chloroplasts, the organelles that power modern plant cells. Again, the pattern was the same: cooperation, integration, specialization. The fossil record is sparse, but there are clues. Microfossils with more complex structures, larger with signs of internal compartments, appear in Proterozoic rocks. Chemical markers, like certain steranes, point to eukaryotic metabolisms. It's not always definitive, but the hints align with molecular data, which suggests that eukaryotes had already split into major groups by the middle Proterozoic. In a way, these cells were worlds unto themselves. They carried within them layers of history, bacterial ancestry, archaeal roots, symbiotic mergers. Their complexity was inherited, not designed. Nothing about it was inevitable, and yet it opened doors that couldn't be closed again. With more energy, more control, and more genetic material, they were positioned to do something new, build multicellular bodies. That wouldn't happen right away. There were still hundreds of millions of years left in the Proterozoic. But the blueprint was now in place. The cell wasn't just a building block anymore, it was a system, ready to cooperate, ready to become something larger. For now, eukaryotes remained small, scattered, and outnumbered. But their presence marked a turning point. Complexity had entered the game, and the next moves would take even longer to unfold. When cells began absorbing other cells instead of digesting them, something strange happened. What was supposed to be lunch turned into partnership. Symbiosis, in the simplest sense, means living together. And in the early Proterozoic, it wasn't always voluntary, but it was effective. One of the most important outcomes of this unplanned intimacy was the mitochondrion, the structure that still powers nearly every eukaryotic cell today. The mitochondrion is often described as the powerhouse of the cell, but that phrase, repeated in textbooks and biology quizzes, barely hints at the story behind it. Mitochondria didn't start as organelles. They began as free-living bacteria, likely a type of alpha proteobacterium. Capable of using oxygen to produce energy efficiently, these bacteria had a metabolism well suited for a planet in the early stages of oxygenation. But instead of continuing on independently, one of them ended up inside another cell, and stayed. How that happened is still debated. Some theories propose that the host cell engulfed the bacterium through a process like phagocytosis, pulling it in as a potential food source. Others suggest a more mutual beginning. Perhaps the host provided protection, and the bacterium offered useful chemical outputs. Either way, the result was not digestion but accommodation. And over time, what started as cohabitation became permanent. The bacterium transferred many of its genes to the host cell's nucleus. It lost its autonomy, its ability to live on its own. It became dependent, and so did the host. The bacterium provided a consistent internal source of adenosine triphosphate, ATP, the cell's main energy currency. In exchange, the host cell gave it raw materials, shelter, and control over its reproduction. This was not a smooth transition. It took millions of years. But gradually the merged entity stabilized, and the mitochondrion became a fixture, a cell within a cell. A biological merger so complete that most modern eukaryotic cells cannot survive without it. Mitochondria still carry traces of their bacterial past. They have their own DNA, separate from the cell's nucleus. They reproduce independently, dividing in a way that resembles bacterial fission. They have double membranes, one likely from their original bacterial envelope, the other from the host's engulfing process. Their ribosomes and some proteins resemble those of bacteria more than those of their host. This cellular fusion marked a turning point in evolution, not because it made eukaryotes more interesting, but because it made them more powerful. Aerobic respiration, the process used by mitochondria, produces many times more energy per molecule of glucose than anaerobic methods. That extra energy meant cells could grow larger. They could maintain complex internal structures. They could copy more DNA, produce more proteins, and perform more functions. Energy made complexity sustainable. The impact of mitochondria isn't just metabolic, it's ecological. Cells with mitochondria could exploit more environments. They could tolerate oxygen and eventually depend on it. They could form new kinds of communities, interact in new ways, build new structures. The quiet absorption of one bacterium into another opened the door to the entire tree of complex life. Some eukaryotes went further. They formed additional symbioses. In lineages that would become algae and plants, another major merger occurred. A photosynthetic cyanobacterium was engulfed and retained. This event, separate from the mitochondrial one, gave rise to the chloroplast, the organelle responsible for capturing sunlight and producing energy from light. Like mitochondria, chloroplasts have their own DNA, double membranes and bacterial ancestry. Symbiosis, once seen as a biological footnote, is now recognized as a driver of major evolutionary transitions. It's not just a survival strategy, it's a creative force. Instead of discarding the foreign, early eukaryotes incorporated it. Instead of rejecting the other, they made it a part of themselves. And in doing so, they rewrote what a cell could be. The mitochondrial origin story also challenges tidy narratives about competition. Evolution isn't only about outcompeting rivals, it's also about integration. The most successful organisms aren't always the fastest or strongest. Sometimes they're the ones that learn to live together. Even now, mitochondrial function affects every aspect of eukaryotic life. From muscle contraction to neural activity, from reproduction to aging, the energy economy of the cell runs through these ancient bacterial descendants. When mitochondria fail, systems collapse. When they're efficient, life flourishes. They are not optional accessories. They are the foundation of our biology. And yet, they're still separate in a way. Each mitochondrion carries a tiny genome, a molecular fossil of a time when it was free. It has no awareness of its past, no memory of the engulfing. But the lineage remains. Every mitochondrion in every animal, plant and fungus is a direct descendant of that ancient bacterial partner, taken in more than 1 billion years ago by a cell that chose cooperation over conquest. In the Proterozoic world, this choice didn't make a sound. It left no visible monument. But it marked the beginning of complexity, of cells becoming more than their parts, of life learning how to expand inward before it ever expanded outward. Somewhere in the latter half of the Proterozoic, the Earth nearly shut down. Not geologically, its core still churned, volcanoes still erupted, and the continent still drifted, but climatically. For reasons that aren't fully settled, the planet tipped into a deep freeze. Ice advanced from the poles toward the equator, temperatures dropped, oceans began to seal over with thick sheets of ice. And for a time, the Earth may have looked more like a frozen moon than the warm, blue planet we recognize today. This wasn't just another cold spell. It was a glaciation of unusual scale and severity. At least twice during the Proterozoic, and possibly more, ice covered much of the planet's surface. Not just the poles or the mountaintops, but low latitudes, places near the equator, shallow tropical seas, entire continents vanished under glaciers. The term that gets used is Snowball Earth, and while there's still debate about how complete the coverage was, there's little question that something extreme occurred. The evidence is in the rocks. Glacial deposits from this time show up in places where glaciers shouldn't have been, if modern climate patterns had applied. Drop stones, large rocks carried by ice, and then released into fine sediment as the ice melted, appear in ancient marine layers, far from today's polar zones. Glacial striations, scratches left by moving ice, show up on exposed bedrock in regions that are now warm. These geological clues point to a world where cold reached into nearly every corner. What could have caused such a deep freeze? The answer isn't simple. Climate is a balancing act between incoming solar radiation, atmospheric composition, ocean currents, and surface reflectivity. During the Proterozoic, the sun was still fainter than it is today, producing only about 94% of its current output. That alone made the planet more vulnerable to cooling. Then there's the atmosphere. Earlier in the Proterozoic, methane was an important greenhouse gas, helping to trap heat. But as oxygen levels rose, thanks in large part to the activity of Cyanobacteria, methane was chemically broken down. Carbon dioxide remained, but if enough of it was drawn out of the atmosphere by weathering processes or biological activity, the greenhouse effect could weaken. That would open the door for cooling. Once ice began to form, it accelerated the problem. Snow and ice reflect sunlight, unlike darker surfaces that absorb it. As more of the planet turned white, less heat was retained. Temperatures dropped further. More ice formed. It was a feedback loop, and once it reached a certain point, it was hard to stop. The result was global or near-global glaciation. What happened to life during this time is harder to determine. Fossils are sparse, and most of the surviving biosphere would have been microbial. Shallow water habitats, the homes of photosynthetic mats and early eukaryotes, were likely the most affected. Ice covered the oceans, blocking sunlight, cutting off the energy supply. Some life may have retreated to refugia. Cracks in the ice, geothermal hotspots, deep-sea vents, isolated patches of open water. Others may have gone dormant, waiting for conditions to shift. But life didn't end. Somehow, it endured. Microbes adapted. Anaerobic organisms continued their quiet work in the sediments. Chemotrophs near hydrothermal vents kept their cycles running. And photosynthesis, though limited, probably persisted in thin layers beneath the ice or in meltwater pools. Eventually, something changed. The cold didn't last forever. Volcanic activity, always simmering beneath the surface, continued to release carbon dioxide. With much of the planet frozen, there were fewer ways to remove that CO2, less weathering, less uptake by plants or microbes. Over time, the gas accumulated. The greenhouse effect intensified. Temperatures began to rise. As the planet warmed, the ice began to melt. Slowly at first, then faster. Oceans opened up again. Sunlight reached the water's surface. Atmospheric moisture increased. The snowball began to crack. This recovery wasn't gentle. The end of a snowball Earth was likely marked by violent climate swings. Rapid warming, acid rain, as the atmosphere reabsorbed volcanic gases, flooding from melting glaciers, nutrient surges in the oceans, triggering blooms of microbial life. The aftermath was a period of environmental instability, but also opportunity. Chemical weathering of exposed land returned. Oxygen levels rose again. Ecosystems reorganized. And in the wake of these extreme shifts, the groundwork was laid for even more complex life to emerge. Snowball Earth events are often framed as disasters, and by many definitions they were. Massive environmental stress, potential mass die-offs, disruption of global cycles. But they also reset the stage. They may have pruned stagnant ecosystems, cleared niches, and forced innovations. In their aftermath, new evolutionary pathways became viable. Not because the freeze was good, but because life adapted around it. The Proterozoic was never a time of calm stasis. It had its quiet stretches, yes. But it also swung through chaos, through global rusting, atmospheric upheaval, and now deep freeze. And each time life held on, sometimes just barely. But it never let go. The cold didn't kill everything. It narrowed the options. It boxed life into corners, but it didn't erase it. Beneath the thick, gleaming armour of global ice, life found ways to hold on. Not through defiance, but through persistence. The Snowball Earth periods of the Proterozoic were among the harshest environmental challenges in our planet's history, but they didn't mark an end. They marked a narrowing of paths, a funnel through which only the hardiest, simplest, or most cleverly situated organisms could pass. The surface of the Earth, during a snowball phase, was largely unrecognisable. Continents were buried under glaciers, oceans were sealed beneath ice sheets that may have been up to a kilometre thick. Winds howled through thin, dry air. Temperatures dropped far below freezing. photosynthesis, the energy engine of most surface ecosystems, was largely shut down. The sunlight that reached Earth was weak to begin with, and what little got through was reflected back into space by ice. But the entire planet wasn't dead. Underneath the ice, the ocean still moved. Heat from the planet's interior continued to warm the deepest waters. Hydrothermal vents, those volcanic openings in the seafloor, still spewed mineral-rich fluids, creating small oases of chemical energy. Microbial life, already long adapted to these extreme environments, continued largely unaffected. These deep-sea communities ran on chemosynthesis instead of photosynthesis. Instead of using light to produce food, they used chemical reactions, oxidizing hydrogen sulfide, methane, or other substances to generate energy. Bacteria at the base of these food webs were efficient, tough, and indifferent to the freezing conditions above. They had their own rules, and those rules still applied. In shallower waters, things were more precarious. Some models suggest that thin cracks or patches of open water, called polynyas, might have existed even during the height of glaciation. Wind patterns, ocean currents, and geothermal activity could have kept small areas from freezing completely. In these slivers of habitability, photosynthetic microbes could still access light. Not much, not for long, but enough to survive. Beneath the ice, there may have been liquid water trapped between the ice sheet and the ocean itself, a slushy, dimly lit boundary where temperatures hovered just above freezing. Microbes here might have clung to the underside of the ice or floated in brine channels. Similar systems exist today beneath Antarctic ice shelves, where microbial life thrives in low light and near freezing temperatures. On land, things were quieter. Without plants, soils, or animals, the glaciated continents didn't support much visible life. But within rocks and beneath the surface, microbes likely endured. Endoliths, organisms that live inside rocks, could survive on minimal resources, shielded from radiation and buffered against the cold. They've been found in similar conditions today in Antarctic deserts and deep underground. They metabolize slowly, dividing perhaps once every few years, but they persist. Other microbes may have entered dormant states. Spores, cysts, and other resistant forms could survive long stretches of inhospitable conditions, waiting for the environment to shift. Some of the earliest eukaryotes might have taken this route, hibernating through the worst of the freeze, conserving their internal structures and reactivating only when light and temperature allowed. This strategy, waiting rather than fighting, was likely a common theme. Evolution doesn't always favour the fastest or the most aggressive. In times of stress, it often favours the smallest, the simplest, the most patient. Cells that could go quiet, that could slow their metabolism, that could endure low nutrient levels for extended periods, they had a better chance of seeing the other side. And when the thaw came, it didn't just bring relief, it brought opportunity. As ice melted and oceans reopened, nutrient-rich waters from deep below rose to the surface. Minerals trapped under glaciers or concentrated in isolated basins mixed into the photic zone where sunlight now reached again. This nutrient surge may have triggered massive blooms of microbial life. Photosynthesis roared back to life. Oxygen levels began to climb once more. These post-glacial conditions were chaotic, fast warming, intense weathering, shifts in ocean chemistry. But for the survivors, it was a renaissance. New energy sources, open ecological niches, fewer competitors. For the eukaryotes that had managed to hold on, whether near vents, in meltwater pockets, or through dormancy, this was their chance to expand. Some of the oldest known multicellular fossils appear in rocks just above Snowball Earth glacial deposits. They're not animals, not yet, but they show a new level of organisation. Cells working together, forming bodies, cooperating in ways microbes rarely had before. It's not that the cold created complexity, but it may have cleared the space for it to spread. Survival didn't mean conquering the ice. It meant adapting around it, shrinking, slowing, hiding, and when the ice retreated, those quiet survivors emerged, changed by the experience, ready to reshape the biosphere again. The snowball didn't erase life, it concentrated it. It tested resilience, and it sharpened the tools life would soon need to grow more complex, more diverse, and far more visible. While life in the Proterozoic was navigating glaciations and atmospheric upheavals, the ground beneath it was never still. The continents, though largely barren and silent from the surface, were in constant motion. Not fast, nothing in geology ever is, but steady, insistent, and far-reaching. Below the microbial mats and frozen seas, the plates of Earth's crust drifted across the planet's surface, pulled by heat from the mantle and guided by invisible currents of molten rock. This movement wasn't chaotic. It had rhythm, a kind of slow, shifting dance that played out over hundreds of millions of years. Tectonic activity didn't begin in the Proterozoic. Plate tectonics had likely been operating in some form since the Archean, but by the time the Proterozoic arrived, the process had matured. Continents had grown thicker, more stable. Cratons, the ancient stable cores of continental landmasses, had formed. These structures resisted erosion and deformation, and over time they collided, rifted, and collided again, building larger landmasses through repeated cycles of aggregation and dispersal. This was the age of the supercontinents. The idea of a supercontinent is simple enough, a time when most or all of Earth's continental land was assembled into one massive landmass. But the reality is less tidy. Supercontinents form slowly, through a series of collisions and sutures, and they break apart in equally uneven ways. During the Proterozoic, multiple supercontinents are thought to have come and gone, each one playing its role in the evolution of the planet. The first major one in this era may have been Columbia, also called Nuna by some researchers. It likely assembled around 1.8 to 2.0 billion years ago, a patchwork of older cratons brought together by subduction zones, mountain-building events, and the slow grind of plate convergence. Its exact shape and extent are still debated, but there's evidence from matching rock formations, mineral belts, and magnetic data that continents we now know as North America, Baltica, and parts of Siberia were once joined. Columbia didn't last. Supercontinents are inherently unstable over geological time. Heat from the Earth's interior builds up beneath them, unable to escape efficiently through the thick continental crust. Eventually, this pent-up heat contributes to rifting. The land stretches, thins, and breaks apart. Magma rises. New ocean basins form, and the continents begin to drift again. After Columbia came Rodinia, forming around 1.1 billion years ago. It was different in configuration, though some of the same cratons were involved. North America, for example, sat near the centre of Rodinia. South America, Africa, Australia, and Antarctica were arranged around it in ways that remain the subject of active research. What's clear is that Rodinia was enormous and probably played a central role in shaping Proterozoic climates and ocean currents. Its formation coincided with massive orogenic events, mountain-building episodes like the Greenville orogeny, which left scars still visible in rock outcrops today. These collisions created highlands, folded rocks, and metamorphic belts, all of which eroded over time to supply sediment to surrounding basins. Those sediments preserved records of the environmental and biological changes that were unfolding. The presence of a supercontinent affects more than just geography. It alters ocean circulation patterns, atmospheric dynamics, and climate. A massive landmass disrupts wind flow, redistributes rainfall, and changes the way heat moves across the planet. Rodinia, once fully assembled, may have contributed to the global cooling that led to the later Snowball Earth events. Its breakup, in turn, would have influenced the recovery by releasing more CO2 through volcanic activity and exposing fresh rock to weathering. Tectonic movement also recycles materials. Subduction zones drag old crust into the mantle, melting and remaking it. Mid-ocean ridges produce new crust. Continents rise, erode, and rise again. Over time, this cycling affects the availability of nutrients in the oceans, things like phosphorus and iron, which are essential for life. The weathering of newly uplifted mountains feeds these nutrients into rivers and seas, potentially stimulating biological activity. The interplay between tectonics and life runs deep. Mountain ranges affect rainfall, which affects erosion, which affects ocean chemistry, which affects ecosystems. Rifting creates shallow seas and continental shelves, which become habitats. Subduction fuels volcanism, which releases gases that reshape the atmosphere. It's all connected, even if the connections are too slow for any single generation of life to notice. From a distance, Proterozoic Earth might have looked quiet. No forests, no animals, no dramatic landscapes in the way we picture them. But underneath, the engine was running. Continents were colliding, scraping, pulling apart. Oceans opened and closed. Mountain chains rose and wore down. And over time, these movements shaped everything from climate to evolution. Even the sky indirectly felt the motion. Tectonics influenced volcanic output, which in turn affected atmospheric composition. Clouds formed differently over supercontinents. Wind patterns shifted. And slowly, life responded. The dance of the continents wasn't graceful. It left scars, faults, and fragments that geologists still trace today. But it was consistent, and it never stopped. The plates kept drifting, the crust kept folding and stretching, and the planet kept changing. Beneath the frozen oceans, above the slow collisions, in the chemical churn of shallow seas, life continued to evolve, quietly, invisibly, waiting for its next big moment. The atmosphere, like everything else in the Proterozoic, changed slowly, not just in composition, but in rhythm. It didn't pulse in hours or seasons, not yet. Not in the way weather drives our lives now. Instead, it moved in drawn-out cycles measured in millions of years, glacial swings, tectonic reshufflings, biological experiments that reshaped its chemistry molecule by molecule. Above the microbial mats and drifting continents, the air thinned and thickened, warmed and cooled, slowly gaining features that would eventually make complex life possible. After the first bursts of oxygen in the Great Oxygenation event, the atmosphere didn't stabilise. Oxygen levels didn't climb steadily to modern values. Instead, they rose, stalled, dipped, rose again. It was less a staircase and more a jagged slope. Oxygen had entered the picture, but it was still just one ingredient in a recipe being revised in real time. For long stretches, oxygen hovered at relatively low levels, higher than the zero of the Archean, but far below what animals would later require. Estimates suggest that for much of the mid-Proterozoic, atmospheric oxygen made up only a tiny fraction of what it does today. Just enough to alter rock chemistry, to rust soils and sediments, but not enough to fuel rapid metabolisms or large organisms. This period is sometimes called the boring billion. Not because nothing happened, but because geochemical records show long stretches of relative atmospheric and biological stability. Not stasis exactly, but slow movement. Life was evolving, the continents were drifting, oxygen was present, but everything felt muted, like the planet was taking a breath that lasted 1 billion years. Yet even in that quiet, important shifts were underway. The carbon cycle, for one, was learning to loop. Carbon dioxide released by volcanoes was pulled back down by weathering. Rain falling on mountains, reacting with silicate rocks, washing into the sea, and eventually getting buried in marine sediments. This process regulates climate. More weathering, less CO2, cooler temperatures, less weathering, more CO2, warmer conditions. Tectonics fed the system by raising mountains, exposing fresh rock. Biology added its touch through microbial activity, influencing the composition of soils and sediments. Methane, once a dominant greenhouse gas, had faded into the background. Oxygen reacts with methane, stripping it from the atmosphere. This change altered the planet's thermal balance. Methane traps more heat than CO2, so its decline helped nudge the planet toward cooling, especially during snowball episodes. The atmosphere became less insulating, more responsive to geological inputs. Ozone began to matter. As oxygen levels rose, ozone formed in the upper atmosphere, creating the first UV shield. This didn't just change what kinds of radiation reached the surface, it shaped where life could live. Before ozone, the surface was deadly. UV rays broke apart organic molecules. Only deep water or shaded microenvironments were safe. But with ozone in place, those protective layers weren't needed as much. New ecological spaces opened up, first in shallows, later on land. Sulfur played a background role. Volcanic eruptions released sulfur gases, which formed aerosols in the atmosphere, reflecting sunlight and cooling the surface. Over time, sulfur cycling became more complex as microbes learned to metabolize different sulfur compounds. The isotopic signatures they left behind in rocks offer one of the best windows into Proterozoic atmospheric chemistry. Nitrogen, too, had its moment. It was already abundant, but biological nitrogen fixation became more widespread. This allowed life to access nitrogen in a usable form, influencing growth rates, biomass, and the overall productivity of microbial ecosystems. In turn, this affected the burial of organic carbon, which fed back into oxygen production. Everything was connected slowly. The atmosphere didn't just change life, it responded to it. Biological processes, especially photosynthesis and respiration, constantly fed and drained gases from the air. When photosynthetic microbes flourished, oxygen rose. When organic matter was buried, CO2 dropped. When tectonic shift brought old sediments back to the surface, previously buried gases re-entered the system. Over and over, the same elements cycled through rock, water, air, and cell. And through it all, the atmosphere gained structure. Stratification began to matter. The troposphere, stratosphere, and beyond. The divisions we know today started to stabilize. Temperature gradients shaped how air moved. Wind patterns emerged. Rainfall began to carve the land in familiar ways. Even without forests or deserts or storms in the modern sense, the air above the Proterozoic Earth began to settle into a more recognizable framework. It was never static. Periods of volcanic hyperactivity would flood the skies with ash and gas. Glacial periods would pull down greenhouse gases and cool the planet. Brief oxygen spikes, like the one at the end of the Neo-Proterozoic, hinted at what was coming, though they didn't last long. Each pulse left a trace in the rocks, oxidized layers, isotope shifts, fossils of microbes adapting to new chemical gradients. And all the while, life remained small. Eyes hadn't formed. Nerves didn't exist. No one could look up and observe the sky consciously. But the air was changing. Slowly, quietly, it was becoming breathable. Not yet, but soon. The chemistry was almost right. The stage was being set. And down below, in the seas and sediments, some cells were beginning to notice each other. Not as competitors, but as potential partners. Not as copies, but as pieces of something larger. It's strange to imagine, but for the majority of Earth's history, life didn't bother with sex. Reproduction, sure, cells divided, DNA copied, offspring produced, but it all happened asexually. One cell became two became four, and the pattern continued. Fast, efficient, and remarkably reliable. For bacteria and archaea, this was enough. Even now, it's how most life reproduces. But somewhere in the Proterozoic, a different strategy emerged. Not a replacement, but an alternative. Messier, slower, riskier. And yet, it changed everything. The arrival of sexual reproduction wasn't a dramatic event. No fossil captures the first time two cells exchange genetic material. No marker in the rock record shouts, "This is the moment." But at some point, eukaryotic cells developed the ability to combine their DNA with that of another, and when they did, they gave evolution a tool it hadn't used before: recombination. In asexual reproduction, offspring are clones. They inherit the full genome of the parent with only minor mutations over time. In sexual reproduction, the story is more complex. Two cells contribute genetic material. Chromosomes are mixed, shuffled, and reassembled in new combinations. Offspring aren't copies, they're variations, and variation, in evolutionary terms, is gold. The benefits of sex from a genetic standpoint are clear. It creates diversity. It breaks up harmful mutations. It allows populations to adapt faster to changing environments. It provides a kind of resilience, a way to explore genetic possibilities more quickly than mutation alone allows. But the costs are real too. Finding a partner takes time. Only half of an organism's genome gets passed on. Mistakes during recombination can be fatal. For sex to be worth it, the benefits had to outweigh the risks. And in the slowly changing chemically dynamic world of the Proterozoic, they likely did. The earliest forms of sexual reproduction probably look nothing like what we associate with the word today. It may have started as simple gene exchange, two cells merging briefly to trade genetic material before separating again. This is still common in microbes, through processes like conjugation, transformation, or fusion. In early eukaryotes, something similar may have evolved. The merging of two nuclei followed by division and redistribution. Over time, these processes became more structured. Meiosis emerged, a specialized type of cell division that reduces the chromosome number by half, allowing gametes to form. When two gametes fuse, the full genome is restored, but with a shuffled combination of traits from each parent. This basic mechanism is now at the core of nearly all complex life. Sexual reproduction likely co-evolved with other eukaryotic features: cytoskeletons, cell signaling, internal organelles. Each new layer of complexity opened up new strategies for interaction, coordination, and reproduction. Some early eukaryotes probably alternated between sexual and asexual phases, depending on environmental conditions. Others may have specialized, developing gametes of different sizes or roles. The first steps toward what we now call male and female. The impact on evolution was immediate, even if subtle. With sex, populations could explore more genetic combinations. Selection could act on new traits more quickly. Beneficial mutations could spread more efficiently. Harmful ones could be masked or eliminated. The genetic flexibility provided by sex helped life weather the instability of the late Proterozoic, freezes, oxygen spikes, tectonic shifts, and come out the other side with more tools in its box. There's also the social dimension, though it wasn't social yet in any real sense. Sexual reproduction required interaction, recognition, compatibility. Cells had to signal to each other, respond to cues, align their timing. This kind of cooperation set the stage for further coordination, something that would be critical as cells began to live not just side by side, but as part of a larger whole. Fossil evidence for the earliest sexual eukaryotes is limited, but there are clues. Some large microfossils show patterns of division and budding that suggest more than simple binary fission. Others resemble modern algae, which often reproduce both sexually and asexually. Molecular clock studies, which use genetic differences to estimate timing, place the origin of sex somewhere over 1 billion years ago, right in the heart of the Proterozoic. We don't know who the first sexually reproducing organisms were. We don't know why they took the risk, but they left a legacy more enduring than any single species. Every plant, every animal, every fungus owes its existence to that decision, to that merger of cells, to that long ago experiment in shared inheritance, and once sex arrived, it never left. Even in lineages that later returned to asexual reproduction, the underlying machinery is often still there, vestigial, dormant, but present. It's a reminder that this wasn't just a feature, it was a turning point. Before sex, evolution had fewer pathways. Afterward, the possibilities multiplied. Not instantly, not explosively, but steadily, quietly. And with each generation, the combinations changed. Life was still invisible to the naked eye. There were no flowers, no mating dances, no parental care. Just cells drifting in ancient seas, trading genes, mixing lineages, setting the stage for a world that hadn't yet imagined animals, or any need for them. Life had always been a solo act. For billions of years, one cell was enough. A cell could feed, reproduce, and adapt on its own. And it worked, still does. Single-celled organisms dominate the planet by numbers, by mass, and by metabolic range. But at some point in the Proterozoic, a few of those cells tried something different. They didn't just divide and separate, they stayed together. They formed clusters, chains, sheets, and eventually something far more cooperative. Multicellularity. It didn't happen all at once, and it didn't happen just once. Multicellularity appears to have evolved independently in multiple lineages, at least a dozen times, possibly more. In algae, in fungi, in slime molds, in animals, and in plants. The idea isn't rare. What's rare is sticking with it, refining it, turning a loose group of cells into a functioning body with division of labour and coordinated development. In its earliest forms, multicellularity probably wasn't much more than sticking together after cell division. One cell divides into two, but instead of drifting apart, they remain attached. Some bacterial colonies still do this. Filamentous Cyanobacteria, for example, form long chains where different cells take on slightly different roles. Some fix nitrogen, others photosynthesize. Even without nuclei, these bacteria were already nudging toward cooperation. In eukaryotes, multicellularity took new shapes. Some cells formed hollow spheres, others developed layers. Some began to specialize. Once specialization starts, the benefits compound. One cell type handles movement, another digestion, another reproduction. The organism becomes more than the sum of its parts, not just a cluster, but a system. But cooperation isn't easy. Cells had to communicate, share resources, and agree, at least chemically, on who would do what. They needed mechanisms to prevent cheating, where one cell might try to take more than its share or reproduce uncontrollably. These early forms of cellular governance eventually evolved into what we now recognize as tissue regulation, growth control, and even immune systems. Some of the first clear evidence of multicellularity in the fossil record comes from Proterozoic microfossils. Not just isolated cells, but arrangements, consistent patterns of size and shape that suggest coordinated growth. Some fossils show structures resembling modern red algae. Others form mats or filaments with cells that appear to have differentiated. They're small, simple, but they're not single. What pushed this shift? Part of the answer lies in energy. The earlier rise of mitochondria allowed eukaryotic cells to generate more ATP than prokaryotes ever could. With more energy, a cell could afford to specialize, to support a neighbor, to participate in something larger. Mitochondria didn't make multicellularity inevitable, but they made it possible. Another factor was the environment. By the late Proterozoic, oxygen levels were rising, slowly but significantly. Oxygen allows for more efficient metabolism. It also supports larger body sizes. With oxygen, cells could grow farther from a surface and still receive enough energy. Before this, most life had to stay small, flat, or loosely connected. Oxygen made internal complexity viable. Predation may have played a role too. The emergence of eukaryotic cells capable of engulfing others, essentially eating, created new evolutionary pressure. Single cells that grouped together may have been harder to digest. Clustering could offer protection, and once the clusters existed, selection could begin to favour more organised, coordinated versions. Development became key. In true multicellular organisms, The body plan isn't just a side effect, it's encoded. Cells know, through chemical gradients and genetic switches, when to divide, where to migrate, what shape to take. This required new layers of genetic control. Regulatory genes, signaling pathways, scaffolding proteins, the molecular machinery of bodies. Not all multicellularity aimed for complexity. Many multicellular organisms stayed simple: filaments, sheets, spheres. But some began to build structures that allowed for movement, feeding and reproduction as a group. They didn't just live together, they functioned together, and once that happened, evolution had a whole new playground. Traits could emerge at the level of the organism, not just the cell. Natural selection could act on body shape, growth rate, reproductive strategy. Organisms could become environments for each other, hosts, symbionts, predators, prey. This shift didn't erase single-celled life, it didn't replace it, but it changed the range of possibilities. Multicellularity allowed life to get bigger, to explore new environments, to interact in new ways. It laid the foundation for everything that would come later. Sponges, worms, trees, fish, mammals, humans. And yet, here in the late Proterozoic, none of that had happened yet. The first multicellular forms were still small, still aquatic, still invisible without a microscope. They didn't swim, they didn't sense, they didn't think. They floated, they grew, they cooperated. And in doing so, they took one of the biggest evolutionary steps in the history of life: the decision not to go it alone. Toward the end of the Proterozoic, something changed. Not suddenly, not in an explosive burst, but noticeably. After nearly 2 billion years of quiet cellular life, the fossil record begins to show shapes, forms, patterns that suggest not just multicellularity, but structure. Organised, differentiated, deliberate. These organisms weren't animals as we know them. They had no bones, no jaws, no eyes, but they were bigger, more complex, and unmistakably alive. This period is now called the Ediacaran, and the creatures that appeared during it remain one of the most curious chapters in Earth's biological history. The rocks that hold their imprints come from a narrow slice of time, roughly 635 to 541 million years ago, just after the last Snowball Earth event. Glaciers had retreated, oxygen levels had risen again, creeping toward thresholds that could support larger bodies. Ocean chemistry had shifted, providing new gradients of nutrients, and in this environment, new forms of life began to occupy the sea floor. Ediacaran fossils don't resemble anything alive today. They were soft-bodied, lacking shells or skeletons. Many had flat, quilted bodies pressed gently against the sea floor. Others had fronds or discs. Some lay still. Others may have waved or undulated. They left behind faint impressions in ancient sandstone, ghosts of living forms, preserved in silt and microbial mats. We don't know what most of them were. Some researchers see them as early animals. Others argue they belong to entirely separate branches of life, now extinct. Some may have been early relatives of cyanidarians, like jellyfish and corals. Others may have been giant single-celled organisms. A few perhaps were colonial. None of them had hard parts. Very few show evidence of mouths, digestive tracts, or sensory organs. And yet, there's order. Some fossils show bilateral symmetry. Others suggest radial patterns. A few seem to have held themselves upright in the water, rooted or anchored to the sediment. Size varied, but some reached lengths of up to a metre, enormous by the standards of the time. They lived on microbial mats, those same layered communities that had dominated for billions of years. But now the mats weren't just the builders, they were the stage. The Ediacaran organisms rested on top of them, fed off them, or maybe absorbed nutrients directly from the surrounding water. In many cases, there's no evidence of feeding structures. They may have relied on osmosis, or absorbed dissolved organics passively. It's hard to say. Some moved, slowly, perhaps by cilia or muscular contractions or coordinated cell movements. Trackways preserved in ancient seabeds hint at motion. Simple, meandering paths pressed into the mud. Not hunting, not fleeing, just repositioning. Looking, maybe, for better conditions. What's clear is that these organisms represent a departure, not just larger, but more organised. Not just stuck together, but shaped. Development, cell specialisation, tissue coordination. These weren't theories anymore. They were living experiments, fossilised mid-step. And yet, they remain elusive. Few show signs of reproduction. Fewer still have internal structures preserved. There are no skeletons, no teeth, no eyes. They don't fit neatly into later evolutionary lineages. Some vanished completely at the end of the Ediacaran. Others may have given rise to the animals of the Cambrian explosion, but the connections are blurry. It's like flipping through a photo album from a lost civilization, recognisable as life, but foreign in every detail. Their world was different too. Oxygen levels were higher than they'd been, but still well below modern levels. The oceans were stabilising after a long period of glaciation, rich in nutrients from weathered rock. Microbial life still dominated, but now there were gradients, light, oxygen, nutrients, that allowed for ecological layering. Competition was still mostly chemical, but interactions were becoming more spatial. Who lived where and in what shape began to matter. Ediacaran communities were not dense. Fossil beds show low diversity, few overlapping forms. Each organism had its patch of seafloor spaced out, undisturbed. It wasn't a crowded world, but it was a more varied one. The biosphere was trying new things, and then, around 541 million years ago, they disappear, not all at once, but gradually. Their forms fade from the rock record, replaced by burrows, tracks, shells, marks of a new kind of life, mobile, active, more complex in function than form. The Ediacarans didn't lose a battle. They may have been outpaced, outcompeted, or simply swept aside by ecological changes they weren't built to survive. Their disappearance is still a mystery, but their presence matters. They mark a transitional moment, when multicellularity stopped being theoretical and became visible, when the rules of evolution shifted again, when form began to follow function, and when complexity, real complexity, started to shape the surface of the planet. They weren't the end goal. Nothing in evolution ever is. But for a brief span of deep time, the Ediacarans were Earth's most elaborate experiment, and in their shadows, something else was getting ready. Something with nerves, with movement, with a new way to interact with the world. The Proterozoic didn't end with a bang. There was no comet, no mass extinction etched into the rock in blackened layers, no sudden vanishing of species across the globe. Its closing act was more subtle, a slow reshaping of the world's conditions, a gradual transition into something new. And for a stretch of time that had lasted almost 2 billion years, that kind of ending was fitting, quiet, deliberate, and absolutely full of change. By the late Ediacaran, Earth was no longer the same planet it had been at the eon's beginning. The atmosphere, once choked with methane and devoid of oxygen, now held enough O2 to support larger, more active organisms. The oceans, once anoxic and saturated with dissolved iron, had shifted toward more oxygenated surface waters, allowing new forms of marine life to emerge and persist. Continental crust had grown and stabilised, shaped by the long cycles of tectonic collisions and supercontinent assemblies. Rodinia had come and gone. Its pieces were drifting, stirring ocean currents, rerouting climates. Beneath the surface, the biosphere had transformed too. Once the exclusive domain of microbes, life now existed in multiple layers. Simple animals were beginning to appear, soft-bodied, hard to classify, but motile. Burrow marks in the sediment suggest that something with a front and back, something that could move with purpose, was now reshaping the seabed itself. The appearance of these trace fossils hints at new kinds of behaviour: grazing, hunting, avoiding. In short, interaction. This alone marked a fundamental shift. For most of the Proterozoic, organisms had reacted to the environment. Now, they were beginning to shape it. Not on the grand scale of forests or reefs, not yet, but the groundwork was being laid. A worm tunnelling through mud may seem small, but it changes how sediment settles, how oxygen penetrates, how nutrients cycle. These changes ripple outward. The ecosystem adjusts. The tools for modern evolution were all in place now. Sexual reproduction had taken hold. Multicellularity was no longer an anomaly. Communication between cells had been refined. Developmental processes were becoming more complex. Bodies were now things that could be inherited, shaped, selected. The rules had expanded. But even with all these changes, the Proterozoic didn't conclude with a sense of finality. There was no moment when the curtain dropped. No day the planet woke up to find itself in a new eon. The transition into the Phanerozoic was gradual, smeared across rock layers and biological innovations that don't neatly align with our human need for clean edges. The boundaries we define, between Ediacaran and Cambrian, between Proterozoic and what comes after, are useful, but they're arbitrary. Still, something shifted. Fossils from just beyond the boundary show new forms, animals with symmetry, with shells, with segmentation, creatures that moved with intention, organisms that fed on each other. The ecological web grew more tangled, more dynamic. Niches filled faster, competition ramped up, and in response, life evolved faster, dramatically faster. The Cambrian explosion, as it would later be called, didn't erase the Proterozoic. It built on it. Every feature that made complex animals possible, oxygen, energy, cooperation, reproduction, multicellularity, all of it had been worked out, tested and refined in those quiet ancient seas. Without the Proterozoic, there would be no vertebrates, no plants, no complexity at all. So as the eon came to a close, the stage wasn't being struck, it was being reset. Organisms that had persisted in low diversity were now poised to radiate. Small changes in form could now lead to large changes in function. The genetic and environmental constraints that had kept evolution moving at a crawl were loosening. Oxygen, finally high enough, made high-energy lifestyles possible. Shells offered protection and structure. Segmentation allowed movement and specialization. Eyes, real, image-forming eyes, were just around the corner. But in this moment, the last breath of the Proterozoic, the world was still half-formed. Microbial mats still carpeted the seafloor. Ediacaran organisms still clung to life in some regions. Oxygen levels, while improved, still fluctuated, and evolution still moved one generation at a time. The silence was deceptive. Underneath it, the gears of biology were accelerating. No one watched this happen. There were no observers, no record keepers, just the fossil traces, the chemical residues, the scattered clues left in ancient stone. And in them, we see the outlines of something familiar taking shape. The Proterozoic didn't end because it was done. It ended because it had done enough. It had built the platform, stabilized the atmosphere, diversified the cell, invented sex, complexity, cooperation. It didn't need to do more. Life was ready to take the next step. The curtain didn't fall, it lifted. We tend to picture evolution as a straight line, a slow march from simple to complex, from bacteria to people. But the truth is far messier. There was no goal, no direction, no promise of intelligence or cities or language. What there was, what there still is persistence, experiment, inheritance, and at the root of all of it, microbes. Everything we are begins with them, not just in the abstract, not just as some distant step on a long evolutionary ladder, but directly, intimately, biochemically. The cells that make up our bodies still carry the architecture of Proterozoic life. Our mitochondria, inherited through countless generations of eukaryotic ancestors, are direct descendants of once-free bacteria. Our DNA replicates using mechanisms first refined in ancient microbial lineages. Our metabolisms run on pathways mapped out by cells that lived in oceans billions of years ago. You don't have to go far back in your lineage to hit microbial roots. Every animal, every plant, every fungus, everything with a nucleus, is part of the same experiment that started when one cell engulfed another and decided not to let go. Everything multicellular came from a choice, made again and again to cooperate rather than divide. And everything complex builds on the infrastructure they left behind. The Proterozoic is long gone, but it never really ended. It's still inside us. The timing mechanisms of our cells, the membranes that separate inside from out, the way we process oxygen, the way we grow, the way we reproduce, all of it shaped by organisms that lived in warm, shallow seas before the continents had settled into anything familiar, before animals had spines, before anything had a brain. We carry their quirks, the genetic code they settled on with its redundancies and workarounds, the cellular compartments they evolved, the tools for copying and cutting and pasting bits of information, even the mistakes, they're still with us. Mutations that slip through, proteins that misfold, aging processes that begin with tiny molecular inefficiencies inherited across eons, and we carry their resilience. Those first microbes endured heat, poison, starvation, glaciation. They retooled their metabolisms, rewired their genomes, survived every mass extinction this planet has thrown at them. Some of them never changed much at all. Others became parts of us. We're not separate from them. We're a branch. And not even a large one. If you tallied up all the biomass on the planet today, the microbes still win by mass, by diversity, by reach. They live in your gut, on your skin, in the soil, in the clouds. They digest your food. They make your vitamins. They train your immune system. You're not just descended from microbes, you're still outnumbered by them. And when we look back at the Proterozoic, what we're really doing is trying to remember something we've forgotten. Not a time before life, but a time when life was something else. Slower, quieter, no less alive, no less important, just different. The silence of that era wasn't emptiness. It was depth. Those ancient oceans weren't lifeless. They were crowded with things too small to see. Crowded with ideas, with evolutionary experiments, with strategies that would echo into the future. Some failed. Many failed. But the ones that worked, energy capture, cooperation, reproduction, communication, they were kept, refined, passed on. You and I are not exceptions to this. We are its outcome. We're not the end of a process. We're part of its continuation. And the rules that shape the Proterozoic world still shape us. We breathe because oxygen became common. We metabolize because of ancient bacteria. We inherit because some eukaryote 1 billion years ago figured out how to recombine chromosomes. We don't live after the Proterozoic. We live because of it. So the next time you look at your hand, or breathe deeply, or think a thought, remember that these things didn't emerge out of nowhere. They were built piece by piece by life that never looked like us, never imagined us, never needed to. The quiet cells that drifted in ancient seas, that layered themselves into mats, that merged and divided and evolved, they're still here, in our bodies, in our blood, in our cells. You're not just from Earth, you're from deep time, and the microbes, they were here first. They built the world, and they brought us along for the ride. That was the Proterozoic, not a highlight reel, not a spectacle, not a chapter most people learn about in school. But it was nearly half of Earth's entire history, almost 2 billion years, and during that quiet stretch, nearly everything essential happened. Cells figured out how to cooperate. Oxygen became something life could use. Continents came together and drifted apart. The ocean froze over, twice. And through it all, microbes worked, adapted, endured. Slowly, patiently, they built the systems that would allow complexity to exist. The Proterozoic wasn't flashy. It didn't need to be. It was methodical, experimental, relentless. It wasn't about what life looked like. It was about what life could become. Without it, there's no us, no thinking, no breathing, no bodies, no language. Every single part of what we call life now is an inheritance from that deep, ancient stretch of time. From the quiet, from the small. We didn't arrive at the end of the story. We arrived long after the groundwork was laid. And that work, hidden in stone, preserved in chemistry, encoded in our cells, is still unfolding. You don't need to go back to the Proterozoic to find it. You're carrying it with you.